Hello, everyone, and welcome to today's webinar, B Cells and Beyond, Understanding Immune Pathways in Kidney Disease. We appreciate you taking the time to join us for this important discussion. Our goal today is to explore the immune mechanisms that contribute to the development and progression of glomerular diseases, with a focus on B cell biology, immune dysregulation, and the pathways that drive kidney injury. We'll also consider how these insights may inform our understanding of disease progression and clinical outcomes. This program is sponsored by Otsuka Pharmaceutical Development and Commercialization. The speakers in today's webinar are employees or paid consultants of Otsuka. To ensure we're aligned from the start, here are a few important guidelines. This is a scientifically rigorous, unbiased discussion focused strictly on disease state education. This webinar does not provide CME credit, and we cannot provide clinical advice on specific patient cases. And finally, this discussion does not promote any specific products or therapeutic agents. With that, let's get started. Now let me introduce myself and today's speakers. My name is Dr. Josh Garibaldi, and I'm a Senior Medical Science Liaison in Nephrology and Rare Diseases with Otsuka. My role is to bridge scientific advances with clinical practice by engaging with nephrology experts and facilitating discussions like today's. Joining me today are two distinguished colleagues. First, Dr. Yelena Drexler, Associate Professor of Clinical Medicine in the Katz Family Division of Nephrology and Hypertension at the University of Miami Miller School of Medicine, where she also serves as Director of Clinical Research at the Katz Family Drug Discovery Center. Dr. Drexler earned her medical degree and completed residency and fellowship training at Columbia University Medical Center. Her work is dedicated to advancing precision medicine to improve outcomes in patients with glomerular diseases. Also with us is Dr. Mark Richards, a nephrologist at Florida Kidney Physicians, where he leads the Glomerulonephritis Center of Excellence in Boca Raton. He earned his medical degree at the University of Miami and completed fellowship training at the University of North Carolina Kidney Center. Dr. Richards has dedicated his career to improving care for patients with complex glomerular diseases, blending academic training with hands-on experience in a specialized clinical setting. We're fortunate to have both of them with us today. Here's what we'll cover today. We'll start with an overview of the immune system, then turn to the role of B cells in immune defense. From there, we'll focus on how B cells contribute to kidney injury, look at specific examples in glomerular disease, and finally discuss the pathways and mechanisms that make B cells so central in nephrology today. Let's begin with a broad view of the immune system, the organs, cells, and proteins that form our body's first defense. Dr. Drexler, could you start us off by describing how these components work together to protect against pathogens?
Absolutely. Thank you, Josh, and thank you to the Florida Society of Nephrology for having us today. So the immune system really operates as a layered defense network. At the most immediate level, we have our anatomic barriers, such as respiratory and intestinal mucosa, which are the first line, preventing pathogen entry at epithelial surfaces. If pathogens are able to bypass these, complement and antimicrobial proteins in the serum, for example C3, are rapidly activated. These serum proteins can directly lyse microbes, serving as an antimicrobial shield at epithelial sites. We then engage the innate immune system. Here, cells like dendritic cells and macrophages provide a rapid but nonspecific defense. We then engage the innate immune system. Here, cells like dendritic cells and macrophages provide a rapid but nonspecific defense. They engulf pathogens, secrete cytokines, and initiate inflammatory cascades that recruit additional effector cells. Importantly, dendritic cells also function as antigen-presenting cells, bridging to the adaptive response. And finally, adaptive immunity, mediated by T and B lymphocytes, provide highly specific recognition through clonal receptor diversity. Although slower to develop, this arm generates antibodies and cytotoxic T cell responses with memory, enabling a more durable and efficient defense upon re-exposure. So each layer is complementary. Barriers limit entry, proteins neutralize early invaders, innate cells mount rapid but nonspecific responses, And adaptive immunity ensures specificity, amplification, and long term protection.
Thank you, Dr. Drexler. Building on that foundation, the adaptive arm provides specificity and memory. And this is where B cells become central players. Dr. Richards, how do B cells generate antibodies, and why is antibody production such a critical part of long term defense?
Yes, thank you, Josh, and to the Florida Society of Nephrology for having me on this discussion. So, B cells are a very integral part of the adaptive immune response. They defend the body by recognizing and secreting antibodies that are very specific to a particular pathogen. And so, B cells actually express immunoglobulins or B cell receptors on their surface that pretty much are made up of a very random array of different combinations of immunoglobulin components. And based on that, that gives them the chance to bind to specific antigens, ultimately leading to activation and proliferation of B cells. This can happen either independently with presentation of an antigen from an antigen presenting cell or with the help of a helper T cell response. When this happens, B cells will mature and differentiate, including something known as class switch recombination. This is where more earlier line B cells, which can express IgM on their surfaces, will then be converted to more specialized types of B cells, specifically IgG, which can help defend against viruses and bacteria in the serum, IgE, which can have a role in the allergic or parasitic response, or IgA, which is primarily the main antibody that is noted in secretions. And this can lead to defenses against viruses and bacteria that can very commonly invade these surfaces. Ultimately, then, B cells can differentiate and convert into plasma cells, which can then release significant quantities of antibodies to bind pathogens and remove them from the circulation.
Thank you, Dr. Richards. So, looking broadly at glomerular diseases, B cells play a role across multiple conditions. Dr. Drexler, what are the main ways B cells drive pathology in diseases such as IgA nephropathy, lupus nephritis, and membranous nephropathy?
Absolutely. So, as you said, Josh, B cells are not a uniform population. They include subsets with very different roles in immune regulation. Effector B cells are pro inflammatory. They drive tissue injury by producing antibodies and cytokines that amplify immune responses. In the kidney, this can translate into autoimmune injury, glomerular inflammation, and even transplant rejection. On the other hand, regulatory B cells, or Bregs, serve as an important counterbalance. They produce immunoregulatory cytokines such as interleukin 10, interleukin 35, and TGF beta. These signals help suppress overactive T cells and promote immune tolerance. In healthy immune responses, that keeps inflammation from overshooting. The challenge, though, in many autoimmune kidney diseases, as well as in transplantation, is that this balance is disrupted. Effector B cell activity is heightened, while Breg function is often reduced or impaired. That imbalance shifts the immune environment toward unchecked inflammation, contributing to disease progression in autoimmune kidney diseases. So, understanding and potentially restoring the balance between effector and regulatory B cells is a key therapeutic target in kidney disease.
Thank you, Dr. Drexler. And so, as you mentioned, when that balance is lost, immune dysregulation follows, and that can have serious consequences for the kidney. Dr. Richards, how does this loss of balance specifically lead to injury, whether from autoantibodies directly or from immune complex deposition?
Absolutely, Josh. So, certainly, the immune system can mediate many forms of renal disease and play a role in the progression of CKD, which is certainly what we are trying to prevent in our patients. And this can be accomplished through either direct or indirect effects. Directly, autoantibodies can be created that target specific renal antigens, and this is very common throughout a host of different glomerular diseases. Ultimately, if these autoantibodies bind directly to glomerular structures, they can recruit complement, lead to further inflammation, and ultimately to worsening glomerular injury, and on the clinical side, progression of chronic kidney disease. Indirectly as well, in the setting of autoimmunity, this can then lead to circulating immune complexes which deposit in different specific structures throughout the kidney based on what glomerular disease is present. At the same time, there are a number of less common diseases that are brought about by genetic defects in the complement system that can lead to a dysregulation of the alternative complement cascade and direct renal inflammation. And there are also a number of hematologic malignancies as well, such as monoclonal gammopathies, amongst others, that can lead to the deposition of specific monoclonal immunoglobulins that led to direct glomerular damage, inflammation, and the progression of kidney disease.
Thank you, Dr. Richards. So, looking broadly at glomerular diseases, B cells play a role across multiple conditions. Dr. Drexler, what are the main ways B cells drive pathology in diseases such as IgA nephropathy, lupus nephritis, and membranous nephropathy?
Yeah, so B cells in particular contribute to glomerular disease pathogenesis through several key mechanisms. One key role is the production of autoantibodies, which can either directly target kidney structures, which occurs in membranous nephropathy, or form circulating immune complexes that deposit in the glomeruli, which occurs in IgA nephropathy and in lupus nephritis. These antibodies and immune complexes then activate complement and recruit inflammatory cells, further driving tissue injury. We know B cells also function as antigen presenting cells, interacting with T cells to perpetuate autoimmune responses and amplify inflammation in these diseases. They also secrete cytokines that shape the local immune environment, further damaging glomeruli and interstitial structures. So, altogether, this dysregulation of antibody production, antigen presentation, and cytokine signaling makes B cells central players in the inflammation and injury that underlie many glomerular diseases. And now that we are armed with a better understanding of disease pathogenesis, it's really become clear that targeting the proliferation, activation, and survival of B cells may improve outcomes and is becoming a very important therapeutic strategy in various glomerular diseases.
Thank you, Dr. Drexler. These diseases share common threads, but B cell contributions look different in each, as you alluded to in this previous slide. But Dr. Richards, could you in more detail compare how B cells are involved in IgA nephropathy versus lupus nephritis and membranous nephropathy?
Absolutely. So certainly, B cells play a very central role in the inflammation in tissue and renal damage that occurs in all three of these specific glomerular diseases. However, there can be some specific differences in their mechanisms of action. Specifically, in lupus nephritis, there is a dysregulation of B cell transcription factors, cytokines, and a B and T cell interaction. There also can be a loss of apoptotic cells that can diminish an overactivated B cell response that can then lead to accumulation of autoantibodies such as double-stranded DNAs that can then proliferate and bind in the kidney and proliferate renal dysfunction. And actually, measurement of these specific double-stranded DNAs is a way to help measure levels of disease activity. In membranous nephropathy, there are circulating antibodies, most notably the antiphospholipid alpha 2 receptor, which can then circulate and bind, specifically in the subepithelial layer near the podocytes, which can then lead to direct glomerular damage, complement recruitment, and ultimately inflammation. In IgA nephropathy, we have what's called the four-hit hypothesis that is very commonly known. And specifically, we have mucosal B cells that produce instead of a regular IgA1 is a galactose deficient or pathogenic molecule that then leads to autoantibody IgG or IgA production against it, formation of immune complexes, and then deposition in the glomerular mesangium.
Thank you, Dr. Richards. So, to understand where these abnormalities begin, it helps to step back and look at B cell development itself. Dr. Drexler, could you walk us through the stages of B cell development and the checkpoints that normally maintain physiologic balance?
Absolutely. So let's drill down a bit into B cell development, which is a tightly regulated process with multiple checkpoints that normally maintain balance between effective defense and immune tolerance. The process begins in the bone marrow, where hematopoietic stem cells differentiate through several precursor stages. These cells then migrate to the periphery, where they can encounter an antigen. Once an antigen is recognized, the B cell can become activated. At this point, activated B cells may undergo class switching to become IgG, IgA, or IgE positive B cells, depending on the cytokine signals they receive. Activated B cells can then differentiate into either antibody producing plasma cells or memory B cells. Plasma cells may reside long term in the bone marrow, secreting antibodies into the circulation, or in mucosal tissues, where they provide local immune defense. Memory B cells, on the other hand, can rapidly redifferentiate into plasma cells upon re-exposure to the same pathogen, ensuring durable immune protection. Overall, this development is dependent on a network of surface receptors and cytokines. CD19 and CD20 are expressed broadly across early and intermediate stages, while CD22 is a marker of mature B cells, and CD38 reaches its highest level on plasma cells. Among the cytokines, BAF, or BLIS, plays a critical role in promoting survival and maturation of B cells in the early and middle stages, while APRIL acts later to support plasma cell survival and antibody production. Altogether, these checkpoints and signals ensure that B cell maturation is both efficient and controlled. But when they become dysregulated, we see the kinds of pathogenic responses that underlie autoimmune glomerular diseases.
Thank you, Dr. Drexler. So B cells don't survive and mature on their own. They rely on these specific cytokines and growth signals. Dr. Richards, what are the key cytokines that support B cell survival, and what happens when those signals are elevated beyond normal levels?
Right. So, as Dr. Drexler alluded to, this is a very, very complex process that involves a number of different regulators, both stimulation and downregulation. And two of the specific proteins that were alluded to are APRIL and BAF, which are significantly key regulators of B cell survival and function. Again, we need B cells. They're essential for both for short-term immunologic responses and long-term immune memory. With the help of APRIL and BAF, we can help stimulate B cells when appropriate. And then ultimately, these can facilitate B cell maturation, differentiation, and survival. So primarily, BAF works in earlier stages of B cell maturation. It can help lead to differentiation and maturation of B cells, as well as proliferation of B cells. Whereas APRIL tends to function a little later in the maturation stages of B cell development. Specifically, it can help regulate the production of activated B cells. It can ensure their survival, and it can also enhance appropriate class switching to whether IgA, Ig, or IgG molecules are needed to mount the appropriate immune response. Certainly, overactivation of these compounds can be quite pathologic and can lead to a number of autoimmune conditions, some of which we had alluded to earlier when discussing perturbations of these conditions in a number of glomerular diseases.
Thank you, Dr. Richards. So here we introduce two of the most important survival signals for B cells that Dr. Drexler and Dr. Richards have already talked a little bit about, BAF and APRIL. Together, they provide critical support for B cell survival and maturation. So Dr. Drexler, could you explain how BAF and APRIL function in this process and what happens when these signals are present at higher than normal levels? I know Dr. Richards talked about that a little bit, but specifically going into how these functions relate to the receptors that they bind to.
Absolutely. So again, as we mentioned, BAF and APRIL are cytokines, right, released by activated antigen-presenting cells, such as dendritic cells and macrophages. And so what they do is they play complementary but very distinct roles in B cell development by binding to specific receptors on B cells. BAF primarily binds to BAF receptor and also engages TACI, with only weak binding to BCMA, which is B cell maturation antigen. BAF receptor is expressed on immature and mature naive B cells, so it promotes survival and maturation during the early stages of B cell development. Now, APRIL, on the other hand, binds strongly to BCMA and to a lesser extent to TACI, but does not bind BAF receptor. TACI is expressed on mature naive B cells and plasma cells, where it facilitates class switching, regulates B cell activity, and supports T cell independent antibody responses. BCMA is highly expressed on plasma cells and is essential for their long-term survival and antibody production. So APRIL promotes later stages of B cell development. Now, when BAF and APRIL are present at higher than normal levels, autoreactive B cells that would normally be deleted can instead persist and expand. This loss of tolerance contributes to autoantibody production, immune complex formation, and chronic inflammation. the processes we've discussed that are central to autoimmune kidney diseases. So, while BAFF and APRIL are indispensable for healthy immune defense, it's dysregulation of these signals that can tip the balance toward pathogenic B cell activity.
Thank you, Dr. Drexler. So now let's take a closer look specifically at APRIL, which plays a particularly important role in plasma cell survival and IgA production. Dr. Richards, what makes APRIL distinct from BAFF, and why is it such a central pathway in diseases like IgA nephropathy?
Absolutely. So, as Dr. Drexler alluded to on the previous slide, BAFF tends to focus more in earlier stages of B cell development and maturation from binding to the BAFF receptor or the TACI. Whereas APRIL will tend to focus more on the proliferation of more mature B cells, as well as involvement with class switching, which can lead to the production of either normal IgA or, in the case of IgA nephropathy, galactose deficient IgA1 molecules. Specifically, APRIL is a cytokine that is released from antigen presenting cells such as dendritic cells or macrophages in response to specific antigens in the circulation. APRIL can then help immature B cells proliferate and survive. And then it can also help, possibly in the setting of T cell stimulation, activate, survive, proliferate, and then differentiate into more active forms. Ultimately, activated B cells can then switch classes with the help of APRIL, as well as ultimately, morph into a plasma cell, which can release significant amounts of IgA or galactose deficient IgA1 into the circulation, or can lead into the production of memory cells, which can sort of be stored to create an adaptive response later in response to a repeat presentation of a similar antigen.
Thank you, Dr. Richards. So here we pulled together genetic and functional evidence comparing BAFF and APRIL, from the GWAS risk alleles to effects on B cell maturation and immunoglobulin levels. Dr. Drexler, what do the GWAS findings shown here suggest about BAFF versus APRIL? And how do those risk variants translate into differences in B cell maturation and circulating immunoglobulin profiles?
Yeah, so genome wide association studies give us important insight into the relative roles of BAFF and APRIL in IgA nephropathy. Interestingly, BAFF risk alleles have not been identified in genetic studies of IgA nephropathy, whereas APRIL risk variants have been consistently associated with disease susceptibility in GWAS studies. That suggests APRIL has a more direct pathogenic role in IgA nephropathy compared with BAFF. As we've seen functionally, BAFF inhibition impacts early B cell maturation, leading to impaired development and a reduction in the overall number of mature B cells. So that translates into a more global reduction in immunoglobulins, including both IgA and IgG. By contrast, APRIL inhibition does not impair maturation. Early B cell development remains intact, but it has a more selective effect at the level of antibody production. Actually, APRIL blockade reduces IgA levels disproportionately compared to IgG. And this is particularly relevant in IgA nephropathy, where galactose deficient IgA1 is the pathogenic antigen driving disease. So the genetic signal and the functional biology really align, indicating APRIL, more than BAFF, appears to be a key driver of galactose deficient IgA production and persistence, making it an especially attractive therapeutic target in IgA nephropathy.
Thank you, Dr. Drexler. So here we go back to the four-hit model of IgA nephropathy, highlighting the steps from mucosal immune activation to abnormal IgA production to immune complex formation and ultimately kidney injury. It also shows where therapeutic strategies are aimed at different points in the process. Dr. Richards, how does this framework help clinicians conceptualize disease mechanisms in IgA nephropathy? And why is it important to think about where different pathways fit into this sequence?
I think this is obviously very important because we now have gone from sort of an old school approach where pretty much our only therapeutic option outside of RAS inhibition were systemic corticosteroids, which sort of just took a blowtorch to this entire four hit cascade. However, now we have much more targeted therapies, which can produce benefits in our patients and result in hopefully much fewer systemic side effects. So we can now focus on all the way at hit four, specifically protection of the glomerulus itself with the help of RAS inhibition, SGLT2 inhibition, reducing intraglomerular pressures, as well as endothelin receptor antagonists, which can play a role in reducing inflammation in the glomerulus as well. We also have complement inhibitors, which have recently been approved for the management of IgA nephropathy, as we know that 90% of cases of IgA nephropathy on kidney biopsy will have evidence of complement deposition. And so certainly this provides an attractive target for protecting our patients against reduction in proteinuria and hopefully reduction in glomerular filtration rates. We can also go further upstream and try to reduce the production of pathogenic galactose deficient IgA1 and all of its potential or downstream effects. Specifically, we can target the Peyer's patches in the terminal ileum with delayed release corticosteroids that can have a less toxic effect than more systemic corticosteroids that were used prior. Or we can even now, as we discussed before, target both BAFF and APRIL with either APRIL inhibitors by themselves or BAFF-APRIL inhibitors, potentially reducing the production of the galactose deficient IgA1 due to less proliferation of these abnormal B cells. As we know, there are increased levels of APRIL that are noted in patients with IgA nephropathy, and so blocking of the production of APRIL might be a very attractive target as well for our patients with IgA nephropathy.
Thank you, Dr. Richards. And thank you, Dr. Drexler. We've covered a lot of ground today, from the basics of B cell biology to their role in glomerular diseases and immune dysregulation. To close this out, let's reflect on the big picture. Dr. Drexler and Dr. Richards, what would you each highlight as the single most important takeaway for clinicians from today's discussion?
Sure. So for me, the key takeaway is that, as we've said, B cells are active drivers of immune dysregulation in glomerular disease. And understanding their biology helps us connect molecular mechanisms directly to the patterns of injury we see in the clinic. And I would also emphasize that this growing knowledge base is not just academic, it's already guiding the development of targeted therapies. And by linking disease mechanisms to treatment strategies, we have an incredible opportunity to move toward more precise and effective care for our patients with kidney disease.
Absolutely. I totally agree. Certainly, I think learning about these topics is very important because we can see potential targets where we can make a difference at the molecular level to then drive improvement in clinical outcomes for our patients, which I believe is the most important thing.
Well, thank you, Dr. Drexler and Dr. Richards, for an excellent discussion. And thanks to everyone watching. Since this program is airing through FSN, a quick reminder that you can also visit nephew.org for additional educational resources, including videos, on-demand webinars, podcasts, and infographics. On behalf of Otska and the Nephew team, we appreciate your time today. See you next time.